students. So students, in the last lecture we have studied about endoplasmic reticulum. So students, we are first studying about all the organelles of the endomembrane system. We know the organelles of the endomembrane system are endoplasmic reticulum, Golgi body, lysosomes and vacuoles, right? So now we are going to study about Golgi bodies, right? The organelle Golgi body was first observed by the scientist. The name of the scientist is Camillo Golgi in the year 1898, right? So the scientist who first observed the Golgi body is Camillo Golgi, and from the same scientist the word Golgi body has came. So the organelle is known as Golgi body from the name of the scientist who first observed it, Camillo Golgi, in the year 1898. Its ultrastructure was proposed by the scientist. The name of the scientist is Dalton, right? So Dalton was the scientist who had proposed its ultrastructure. So students, there are different names of Golgi bodies, right? We are going to study it, right? So Golgi body is also called as Dalton complex, and now you know why it is called as Dalton complex, because the scientist Dalton had discovered its ultrastructure. It is also called as idiosome. Golgi body is also called as lipochondria. Golgi body is also called as lipochondria, and it is also called as dictyosomes, right? Golgi body is also called as dictyosomes. What are dictyosomes? Dictyosomes are nothing but Golgi bodies, right? But small Golgi bodies. So let me write small Golgi bodies in plants are called as dictyosomes, right? So small Golgi bodies seen in plants are called as dictyosomes. It is also called as Dalton complex. It is also called as idiosome. It is also called as lipochondria, and it is also called as Baker's body, right? So these are the other names of Golgi body. One more thing you have to learn is zone of exclusion, right? What is zone of exclusion, right? So Golgi body is an organelle, right? So the cytoplasm which is surrounding the Golgi body organelle is clear where no other organelles are present, right? Cytoplasm surrounding Golgi body is clear where fewer or no other organelles are present, right? And that is called as zone of exclusion, right? So what is zone of exclusion? The clear cytoplasm surrounding Golgi body where fewer or no other organelles are present is called as zone of exclusion. Bigger Golgi bodies are seen in secretory cells, right? Example, the cells of liver and pancreas, and smaller Golgi bodies are seen in non-secretory cells, right? For example, muscle cells, right? a larger sized Golgi body is seen in secretory cells, right? The cells which have to continuously produce and secrete some or the other thing, whether it is hormones, whether it is enzymes, or whether there are some other substances, such as the cells of liver and pancreas. And smaller Golgi bodies are seen in the non-secretory cells, such as the muscular cells. Yes, students, this is clear, right? So, Golgi body was first observed by scientist Camillo Golgi in the year 1898. Its ultrastructure was identified by the scientist Dalton. That is why it is also called as Dalton complex. It is also called as idiosome. It is also called as lipochondria. It is also called as dictyosomes. Very important. What is dictyosomes? Dictyosomes are nothing but Golgi bodies, small Golgi bodies which are seen in plants. It is also called as Baker's bond. Zone of exclusion, right? So the clear cytoplasm which is surrounding the Golgi body, where fewer or no other organelles are present, that area, that zone is called as zone of exclusion. Bigger Golgi bodies are seen in secretory cells and smaller Golgi bodies are seen in non-secretory cells. Yes, students, now we are going to study the structure of Golgi body, right? So, what is the structure of Golgi body, right? Golgi body is made up of 4 components. Structure of Golgi body, right? It is made up of 4 components. Which are those 4 components? The first one is called as cisternae, right? Singular form is cisterna, right? So this you have studied in endoplasmic reticulum also, right? So what is cisterna? It is flat, right, sac-like structures arranged in stack pile, and those are unbranched, right? So cisternas are present. Then the second component is tubules, right? Those are branched. Tubes, right? The third component which is present is vesicles. These vesicles are also of 2 types, that is transition vesicles and dispatching vesicles, right? And the fourth is vacuoles, right? So there are vacuoles also present, right? So these are the 4 components of Golgi body. It is made up of 4 components: cisternae, which have flat sac-like structures; tubules, which are branched; vesicles, transition and dispatching vesicles; and vacuoles. We will draw the structure of Golgi body and then you will understand it in a better way, right? So say we are drawing a cell, we are drawing a very huge cell so that you can understand it properly. Let me So this is a cell, right? Say this is the nucleus of the cell, right? This is the nucleus of the cell. Now Golgi body is having 2 phases, right? There are 2 phases, right? That is cis phase, also called as receiving department also called as forming phase. And the second phase is transphase, also called as shipping department, also called as maturing phase. Right? So there are two phases. Cis phase. Cis phase is convex side and trans phase is concave side. We will understand it once we draw it. So this is the nucleus of the cell. Right? So say this is the nucleus of the cell and here is the endoplasmic reticulum. So this is the endoplasmic reticulum. We have studied the structure of endoplasmic reticulum, so now I am not explaining it precisely, right? So this is endoplasmic this is the connection. Whether it is smooth or rough doesn't matter. Right? We are going to draw the Golgi body. Now, first and the most important component is cisterna. Right? So, say this is one cisterna. It is again a flat and sac-like structure with its ends which are swollen. Right? So, its ends are swollen. Right? So, this is cisterna. Let me draw one more cisterna. Right? This is the third cisterna. This is the fourth one and so on. And its ends are swollen. Right? These cisternas are connected with each other by the branching. Right? So these are the cisternas. So this structure is cisternae. Right? These swollen ends are called as Golgian vacuoles. Right? Swollen ends are called as Golgian vacuoles. So you can see this is the convex side and this is the concave side. So which phase is convex side? It is cis phase. Right? So this part is called as cis phase and this part is called as trans phase. Right? Cis phase is the receiving department. Trans phase is the shipping department. Now there are two types of vesicles which are present. Right? Transition vesicles are the small vesicles which are present towards the cis phase. And the dispatching vesicles are the vesicles which are present towards the trans phase. Right? So these vesicles are transition vesicles and these vesicles are dispatching vesicles, right? And apart from it, there will be also the tubules which are present, right? So there will be tubules also which are present. So now all the structures of Golgi body are there, right? Cisternae which are flat sac-like structures, vesicles. The vesicles which are present towards the cis phase are transition vesicles and the vesicles towards the trans phase are dispatching vesicles. What is cis phase and what is trans phase? Right? The convex side of the Golgi body is called as cis phase. Right? So cis phase is nothing but the convex side and the concave side is called as, so this is the concave side. So it is called as trans phase. Cis phase is receiving department or forming phase. Trans phase is maturing phase or shipping department. Why it is called like that? We will study it right now. Yes, students. So we have studied the structure of Golgi body. We know that it consists of convex side, which is cis phase, and concave side, which is trans phase. Now, why we have given these names? It is called as receiving department or forming phase, and trans phase is called as shipping department or maturing phase. Why? Students, the most important function of the Golgi body is modification and packaging of the materials. Which materials, right? It is modifying. Say for example, there is some raw material, right? If cotton is a raw material, when we modify it and when packaging is done, what we get? We get proper clothes. We get pieces, cloth pieces. Similarly, if anything is produced in the cell which is need to be modified and packaged, that function is done by Golgi body. Say we are talking of endoplasmic reticulum. We know smooth endoplasmic reticulum function is lipid synthesis and rough is protein synthesis. So what happens, students, is that say this endoplasmic reticulum is producing proteins and lipids, smooth and rough. The proteins and lipids which are produced by this endoplasmic reticulum, right, will be packaged in the vesicles. These vesicles are going to travel towards the Golgi body. Now, where it is going to reach? Which part of Golgi body? So, these vesicles are going to reach towards the cis phase of the Golgi body. That is why we call it as a receiving department. It is receiving the materials from the endoplasmic reticulum in the form of vesicles which are needed to be modified and packaged. So, this vesicle, say this was a vesicle which was filled with protein, right? So, it is a protein vesicle. This vesicle has reached the cis phase, that is receiving department. These vesicles are now called as transition vesicles. Now, this vesicle is going to fuse with the cisternae and the material is going to enter inside the cisternae. Now, within the Golgi body, this material is undergoing modification and packaging, right? Say for example, this is a protein and we want glycoprotein. Glycoprotein is nothing but a carbohydrate plus protein. So, carbohydrate is to be added to protein. That modification is done within Golgi body, and finally this vesicle, which is a modified vesicle, is released from the trans phase. That is why trans phase is called as shipping department, and the vesicles are called as dispatching vesicles, or those are also called as discharged vesicles. Are you getting it? So, from endoplasmic reticulum, a vesicle filled with protein has reached Golgi body towards the cis phase. Now, these vesicles are called as transition vesicles. This transition vesicle fuse with the cisternae of the Golgi body and the materials enter inside the Golgi body. These materials undergo modification in the Golgi body, and from this protein, what is formed? Right? So, what does this dispatched vesicles possess? This dispatched vesicles possesses glycoproteins. Right? So, from proteins, this was from ER. Glycoproteins are formed, and that function is done by Golgi body. So now are you getting it? What is the function of Golgi body? Golgi body's main function is modification and packaging of the materials. That is why this phase is forming phase or receiving phase, and from here the vesicles which are dispatched these vesicles might bind with the plasma membrane and the content may be poured outside. That process is called as reverse cytosis, right? So now we are going to study about the functions of the Golgi body. Right? So what are the functions of Golgi body? The first and the most important function is modification and packaging, right? Modification and packaging of the materials. It consists of 3 steps, right? Step 1 is to receive the material from ER through vesicles. Step 2 is modification. That modification is called as glycosylation. What is glycosylation? Addition of carbohydrates. It's called as glycosylation. Addition of carbohydrates. So say for example, if this is a protein and if a carbohydrate is added, then what we are going to get? We are going to get glycoproteins. Glycoproteins are formed, right? If lipids are there and if carbohydrates are added, then what is formed? Glycolipids are formed, right? So this processes are called as glycosylation. These two processes are called as glycosylation, right? So what happens? First, Golgi body is going to receive the material from endoplasmic reticulum in the form of vesicles. Then that material is modified. That modification is called as glycosylation, right? That is addition of carbohydrates. Protein and carbohydrate is glycoproteins. Lipids and carbohydrate is glycolipids. That function is done by Golgi body. Then it also performs the function of liposylation, that is addition of lipids. So say for example, if proteins are there and if lipids are added, what we are going to get? We are going to get lipoproteins. If phosphates are there and if lipids are added, what we are going to get? Phospholipids. That process is called as liposylation, and that is also the function of Golgi body. So finally, what is Golgi body making? It is making glycoproteins, glycolipids, lipoproteins, phospholipids, all these modified structures. It is making enzymes, it is making hormones, right? And finally, the third step is release of modified material through discharged vesicles, right? So finally, once modification is done, it is released by the discharged vesicles. So this is the most important function of Golgi body, that is modification and packaging. Golgi body is involved in the synthesis of glycolipids, glycoproteins, lipoproteins, proteins and phospholipids. Right? The second function of Golgi body is secretions. Right? So second function is, let me write the first function as it is, modification and packaging. The second function is secretion. What is it secreting? It is secreting hormones. Right? It is secreting polysaccharides. It is secreting enzymes. It is secreting proteins. Right? Then the third function is formation of cell plate or phlegmoplast. Right? So this you have studied when you studied cell wall. We have studied that when cell wall is formed, first a cell plate or phlegmoplast is formed, and which organelles are involved? Golgi body and endoplasmic reticulum. The vesicles of Golgi body is forming a cell plate, right? So say this is a cell, right? Then the vesicles of Golgi body is forming a cell plate, and finally it is bursting to release calcium or magnesium pectate, and middle lamella is formed. Then the fourth function is formation of Vitiline membrane of egg. This you will study when you will study the reproductive system, that the ovum is consisting of a membrane. That membrane is Vitiline membrane, and Golgi body is involved in the formation of Vitiline membrane. Then fifth function is formation of acrosome of sperm. Right? So in case of sperms, right, there is head, middle piece and tail portion, and its anterior part is a cap-like structure. That structure is acrosome. This acrosome is filled with many enzymes such as hyaluronidase enzyme. That acrosome of the sperm is formed by Golgi body. Then sixth function is formation of lysosomes. It is also involved in the formation of lysosomes. Endoplasmic reticulum and Golgi body both are involved in the formation of lysosomes. Golgi body is also called as middleman of cell, or it is also called as director of macromolecular traffic. These are the other names of Golgi body. It is also called as middleman of the cell or director of macromolecular traffic. So all of these are the functions of Golgi body. The most important function is modification and packaging. Golgi body is involved in formation of glycoproteins, glycolipids, phospholipids and lipoproteins. It is involved in secretion of hormones, enzymes, proteins and polysaccharides, formation of cell plate or phlegmoplast, formation of the Vitiline membrane of the egg, formation of acrosome of the sperm and formation of lysosomes. Right? So any raw material from endoplasmic reticulum will be received by the cis phase of Golgi body in the form of transition vesicles. Modification is done and finally it is shipped outside by the discharged vesicles through the trans phase or maturing phase. And that is why we can say that the function of Golgi body is reverse pinocytosis. Pinocytosis is cell drinking, right? If there is something over here and if cell want to take it, right, then what happens? There is invagination, right? Material enters inside and then there will be formation of again plasma membrane and this material will be dispatched. This is pinocytosis. What is reverse pinocytosis? Reverse pinocytosis means that this vesicles released by Golgi body is releasing the substance outside. That is why it is called as reverse pinocytosis. In our next lecture, we are going to study the third organelle of the endomembrane system, that is lysosome.